Thanks, Tim. Nice to be here. If we could go to slide 3, please. So Imagion Biosystems is a clinical stage medical technology company. We're focused on improving the way we can detect cancer and other diseases. Our proprietary MagSense technology is non-invasive, doesn't use radioactivity, and provides for a more specific and sensitive detection of cancer than current conventional imaging methods do. We have a large pipeline of products that are already in, some of which are in the clinic as well, which I'll share with you today. Our first product for breast cancer, we've reported some results for just very recently, so I'll share that today. Next slide, please. So we have lots of ways already to image the body, five basic ways to image the body. And so the question really is, why are we focused on medical imaging? And the problem that we really are looking at is that even though we have multiple ways to image the body, there's still an unmet need associated with diagnosing cancer. And that's because these imaging technologies that exist today are good at looking at anatomical problems, such as a broken bone, for example, or blocked arteries, but they can't tell you if the growth they see is actually a tumor or not. We're all familiar with a mammogram seeing a spot, but is it an actual cancer or is it a more benign form of a tumor? So if you go to the next slide, Imagion is trying to change that. Instead of using imaging to only identify a suspicious lesion or a region of interest, our goal is to then actually be able to say that we can detect the tumor cells themselves. So when you get an MR scan, you don't just say there's a region there that's of concern, but we're actually able to point out and say we're able to see cancer cells. Next slide. So how do we do that? So we use tiny magnetic particles that are coated with a targeting molecule, such as an antibody or a peptide, that's highly specific for the type of cancer we want to look for. So for example, if we want to detect HER2 breast cancer, we use an anti-HER2 antibody. If we want to detect prostate cancer, we'll use a peptide that's highly specific for the prostate-specific membrane antigen. And the idea then is that these particles will circulate and seek out the type of tumor cells that they're specific for. And if they find the cancer, they'll bind to the cancer cells, and we'll be able to then detect them in an MR scan. But if there's no cancer there, the particles will harmlessly circulate, they'll get taken to the liver, and then they'll be metabolized normally. And in fact, even the iron that we use is converted into ferritin, which is reused and repurposed for your blood system. So this is a very safe way to be able to use an imaging agent to tag cancers and have them detectable by MRI. If you go to the next slide, please. So what's nice about this approach is this use of targeted imaging can be used with a variety of different types of solid tumors. And we can use conventional MR scanners to be able to detect their presence. So we currently have R&D programs looking at most of these types of solid tumors because we have very good way of knowing how to target these types of cancers. And we know there's clear unmet medical need for all of these. Lung cancers, for example, are notoriously difficult to get biopsies from. Ovarian cancer is very difficult to identify at an early stage because transvaginal ultrasound, which is the conventional way to look at it, can't see ovarian cancer until you have about 10 billion cells and the patient is already stage 3 or stage 4. So in each of these cases, if we can develop an imaging agent that's highly specific for the type of cancer, we can improve on the way today standard of care tries to diagnose cancer. Next slide, please. Our lead product is for the detection of HER2 breast metastasis in breast cancer. It's in a phase 1 study being done in Australia. The imaging agent is used to detect whether or not the lymph nodes have been involved with tumor cells. This is an important medical problem because even though you may have had your primary tumor diagnosed, the first thing the doctor wants to know is, has that tumor spread into the lymph nodes? And unfortunately for patients today, in order to be able to know that, we have to do biopsies on lymph nodes. And there's a lot of morbidity associated with removal of lymph nodes. So if we can non-invasively detect whether or not a patient has nodal involvement, we can improve patient care and patient management, save a lot of morbidity and healthcare costs by being able to not have to use radioactivity, not have to do a biopsy, but still be able to get an answer as to whether or not a patient should be concerned about metastatic disease. We reported the results from our phase 1 study in December at a breast cancer conference, and most recently also reported that we had a blinded review of our study results with a panel of radiologists who've confirmed, in fact, that our findings are that we can use this imaging agent to detect nodal disease for breast cancer patients. Next. So getting this independent confirmation was really an important step for the company because we now have confidence in the data and that we can move on to the next phase of our trials. It means we have a chance to really change the way we can use imaging to detect cancer. Instead of only using imaging to look for morphological characteristics, such as is the node enlarged or is it irregular in shape, we can actually use this change in contrast to determine whether there are cancer cells there. On this slide, you can see, for example, there's a darkening of the node after the imaging agent is given because the patient now has the nanoparticles bound to the tumor cells that we're able to detect in the lymph node. And this change in contrast is what provides the added context for the radiologists. So instead of looking at this image and concluding that this patient has only an enlarged or irregular node, we can now say that the particles are there and they're there because there are cancer cells there. And this could eliminate a biopsy for a patient. Next slide. For each type of cancer, we'll have a unique injectable imaging agent. So this basically, we build a consumable line of imaging tests that are available for different types of cancers. And that imaging test then is sold to the hospital pharmacies and would be requested by the medical oncologist wanting to do the MagSense test. These will be high gross margin consumable products. And our long-term goal is to partner with someone in the imaging space, not to build our own global sales organization, because there are plenty of companies that are already making imaging equipment and such contrast agents. So our goal is to partner with somebody to bring these into the market. We'll be the company who's developing the portfolio of imaging agents and then sharing in the revenue stream associated with the sale of our imaging agents. Next slide. So our business strategy is pretty straightforward. Use our first product for HER2 breast cancer as the commercial launch pad, expand our pipeline of products. So we already have research being done in prostate cancer and ovarian cancer in particular. As I said, we'll be reporting some of our preclinical work in ovarian cancer a little bit in Q2 of this year. Identify those collaborators that are going to help us commercialize the product, and then generate revenue, high gross margin revenue, from those collaborations with the commercial partners. Next slide, please. A quick financial snapshot there. We're a fairly widely retail held shareholder base. We're just getting ready to report our annual results, will be next week, so I won't spend a lot of time on these slides, on the number from this slide, but suggest that you watch for our annual report coming out next week. Last slide, please. So if I were to try to summarize where I think we are, as a company developing medical imaging technology, we've really recently hit some very important milestones. Reporting our clinical results from our first product and having a pipeline of products that we can now start to bring forward towards more clinical testing, I think is really important. And we've got some intellectual property associated with our nanoparticle technology and multiple revenue opportunities associated with that. We've got a very strong leadership board. If you take a look at our board of directors, non-independent directors, all come from this industry. So we have got good leadership there and building a good clinical team to bring these products to market. And with that, I'll be happy to answer some questions.
Thanks, Bob. Quite a few questions coming through. It seems so simple. I actually had an MRI during the week and I was surprised that nothing has changed. It's still a horrific process 10 years later since between MRIs. Has nothing changed in the imaging industry to help identify cancer?
Yeah, so over the years, the imaging technologies have gotten more sensitive, but they are still very focused on anatomical or morphological characteristics. They see an image, but they can't tell you what that actually is. And so the problem with cancer is it's a cellular phenomenon. It's not an anatomical problem. And so they haven't been able to bridge that gap. And so that's really what we're bringing to the table, is we're bringing that molecular answer to the imaging world. And I think that that's going to really be transformative, because PET imaging has started to move in that direction. PET tracers are now starting to be more molecularly specific, but a PET tracer requires a dose of radioactivity. So we think by using this more biologically safe approach of magnetic particles with conventional MRI, we can do a better job of being able to use imaging to detect cancer.
And can you give us a little bit more color on specifically how your nanoparticles actually attach themselves to the cancer cell in layman terms, if you don't mind?
So, no, it's right. So this is what's kind of fascinating about this is this ability to use something like an antibody or a peptide to target the cancer cells has been being used for decades, not in the body, but in the Petri dish. This is exactly how a pathologist stains for detecting whether or not it's a cancer cell. They use an antibody that's labeled typically with a fluor or a stain of some kind. We've just taken that same approach of having an antibody or a peptide that's highly specific for a cancer marker and labeling it with a magnetic particle that's detectable non-invasively through MRI. So it's using relatively known biology where we have antibodies or peptides that are specific for cell surface markers of various types of cancers, but using a way to detect it non-invasively instead of in a Petri dish.
And is there any further, is there FDA approvals or anything like this needed to, any further steps to take in that regard?
Yeah, absolutely. So these are all highly regulated products, just like drugs are. And so we're in the process, our phase 1 study has been being done in Australia. We're in the process now of moving towards application to do the similar kinds of studies in the United States through the FDA. So these will require some further studies and investment so that you've taken enough patients to have statistical relevance to be able to say that, in fact, you can detect cancer when it's there. So it's a normal regulatory process, but it's a little bit different than with a drug because we're not trying to cure the cancer, we're trying to detect it. So the level of efficacy that we need is a little bit different, but we do have to go through the regulatory process, and we're in the middle of trying to start to do that with the FDA.
And you kind of touched on intellectual property. Is there patents involved in your product?
Yeah, so we have some patent applications that we've been putting through that related to this now detection by MRI. We have a library of patents that protect the particle use for a different way of detecting the particles, but we now have to start to build a portfolio associated with MRI. But we think there's some opportunity there to protect this portfolio of imaging agents through both patent and through sort of first mover advantage in the marketplace.
And looking forward, what sort of milestones do you have to achieve, and how do you get to the pathway for sales?
Yeah, so the next major milestones, I think, will be bringing the same study to the United States for the next phase of clinical testing. Another key milestone will be getting a second product into the clinic because we know that we want to attract a strategic partner. And we don't want to be a one-trick pony only with breast cancer. We know that in order to attract a strategic relationship, we want to have a portfolio. So I think key milestones for investors to watch for is our progress in moving this first product through the clinical testing, getting another product into the clinical phase of testing. And it's probably, by getting through the second phase of clinical testing is when we'll be able to start to have a better look at when we'd be able to commercialize this.
And when you talk partnerships, everyone assumes big pharma. Are you talking big pharma, or is there other partners out there?
So there's probably 40 or 50 companies that are in the imaging space, companies like GE, Siemens, Toshiba, Guerbe. There's a large number of companies in addition to the pharmaceutical companies that are specialized in imaging. And so there's a large number of companies that would be interested in what we have, because today, one of the things we're very proud of is that this is the first time that anyone has taken a magnetic particle and dosed a human subject for detecting cancer. This is a first of its kind study. And so we think that we're really bringing something new to molecular imaging, and that would be something that imaging companies don't yet have in their portfolio.
Makes perfect sense. Bob, thanks for your time. I know you got to jump on a plane. We'll finish it up there. Have a nice weekend.
Thank you very much.